Good morning, everyone. Welcome to another lecture from the topic of genetics and molecular biology. And today we'll be talking about the very favorite protein synthesis. When we are talking about the protein synthesis, of course, we are talking about the translation part. Now, in previous lectures, what we have done, we had already discussed about the DNA, the mode of replicating the DNA, the repair pathways, the transcription process, the processing that involves after transcription. That is, the steps which are required to form a mature mRNA or RNA or tRNA. Now, today we'll be discussing the last segment of that central dogma, that is protein synthesis, and why we studied so much about transcription and what is the terminal product, how it is formed, actually how a protein is formed is the main objective of this particular lecture. So the main objective of this lecture is to make you understand, the complete process of translation, how the ribosomes will be coming about to bringing about the translation process, why they are called as a site, or a manufacturing unit for a particular protein. Then if we are talking about the translation process, what role does tRNA is making, how that tRNA is managing as an adapter molecule, how it is bridging the amino acid pool that is present in the cytoplasm, and how it is reading the mRNA and thereby bringing the correct amino acid, and then how the peptide bond that is required for joining the individual monomers of amino acid to form a polypeptide, how that peptide bond is formed, and whether there's a requirement of any energy, is there any energetics that is involved in this particular reaction. So our I expect that by the end of the lecture you will be well versed with all the players that are involved in translation, as well as the complete chronological event, how a single amino acid is added, how the things are initiated, and then how the chain grows, and then finally how a complete translation process in prokaryotes is winding up. So today we'll be focusing only on prokaryotic part. Maybe in the next lecture we'll be focusing on the eukaryotic part. So let us begin. Let us begin with the general brief. See, initial slides. Why I have incorporated these initial slides, just to give you a recap, just to give you a preamble that when we are continuously diving into a new process, that is translation, first let us brush up what exactly we had studied and what we are talking about now. So we all know that what is central dogma, right? It's a flow of genetic information from DNA to RNA, RNA to protein, and then in between the protein, I meant that first a polypeptide chain is formed in the form of primary structure, and then it is folded to give you a three-dimensional conformationally active protein, which actually performs functions. So I'm not talking about the post-translational modifications in this particular category. I'll be only focusing on the synthesis of amino acid chain, and that is translation, right? That is translation. So we'll be talking about this particular step from RNA to protein. So when I'm talking about from RNA to protein, till now we have read that what are the players that are involved in this process, what are the major players. There has to be a template which will provide the genetic information, and that template is mRNA. There is a molecule which will read this mRNA, what is written here, and simultaneously it will bring the amino acid to the site of synthesis, and this particular molecule is called as tRNA. Now, where is the site inside a cell where these two will be going and interacting? That particular site is called as ribosome. This is ribosome where these interactions are taking place. So we understand it very clearly that all the three types of RNA molecules, rRNA in form of ribosome, tRNA in form of carrier molecule, adapter molecule, and mRNA in form of the coding molecule, is very, very essential for protein synthesis. And these are the three players that we had earlier discussed in detail. Now, similarly, if you are talking about the components of translation, what are the different components of translation? See, we had already discussed about the mRNA, the tRNA, the ribosomes, but is this reaction or the formation of bond is happening spontaneously, or it is catalyzed by a certain set of enzymes? So of course, it's a biological reaction, and it is being catalyzed by an enzyme. And the enzymes which are responsible, we'll be discussing in detail that there are a plethora of enzymes which are responsible for executing the translation process. And not only enzymes, there are multiple proteinaceous factors also that are involved. Like at initiation stage, there are initiation factors which are of very, very important, which are of very important. they are actually responsible for, you know, any kind of impairment if they are not performing well. So the initiation factors are involved. Then when the polypeptide chain is growing and the ribosome is moving, that elongation factors are involved. And then when we are talking about the termination, the last state, in that releasing factors are involved. So what are all these factors? These are the protein molecules. These are the protein molecules which are assisting the complete translation process, and in turn these molecules are responsible for maintaining the specificity of the initiation, to maintaining the fidelity of the protein synthesis process. Yes, these proteins are very much essential. So with these players, let us first see what in prokaryote I am talking about in terms of ribosomal RNA. What is ribosomal RNA? We had already discussed this in the previous lecture. So what is ribosomal RNA? See, in prokaryotes, it is of 70S. Now the 70S ribosomal RNA, ribosome unit, the 70S ribosome unit is made up of two units, large and small. You can see in the picture, it is the one is large and the other is small. The cumulative weight is 25 lakh molecular weight. If you'd say, if you split it into two parts, then there will be 16 lakh and the 9 lakh molecular weight. With 50S, S means Sidberg unit. We are talking about the rate of sedimentation. Then it splits into 50 and 30. Now, please, students, remember it clearly. I am again emphasizing on this particular part. 70S, if we split it into a large and small subunit, it is 50 and 30. Mathematically, 50 plus 30 is 80, but not biologically. In case of ribosome, it is 50 and 30 is giving rise to a 70S, because we are not talking about mathematical addition, we are talking about the rate of sedimentation, and when you are combining it, it is giving you a 70S representation. Fine. So in prokaryotes, the large subunit is 50S and the small subunit is 30S. And in this large unit, we have 34 different proteins bind to two different ribosomal RNA, that is 5S RNA, which is the smallest one, and the 23S RNA, which is made up of 2900 nucleotides. Whereas the small subunit, which is 16S rRNA, this unit is the one of the, it's the main player in my opinion in starting the translation process, because it first recognizes the mRNA, it first recognizes the correct position on the mRNA, and then it starts the initiation process, and then later on the larger assembly is coming. So we'll see that process in just upcoming slides. So we have to remember that the smaller subunit is of 30S, and in that 30S there are 21 different proteins, but there is only one ribosomal RNA, that is 16S rRNA. 16S rRNA. Then when we talk about the eukaryotic S, this is the picture, this is the bifurcation, but we will be focusing this when we are talking about the eukaryotic translation. Then what about tRNA? What about tRNA? We had already studied this, so I'm not going in detail with the cloverleaf model. I have just showcased this particular slide for two different reasons. The first reason is the anticodon loop, and the second reason is to remind you that there is a CCA, the CCA, three nucleotides that were added by terminal nucleotide transferases. So at the 3' end of a tRNA, there is a adenine nucleotide with the base of adenine, and we have a 3' OH free. We have a free 3' OH. Now, why I'm telling you? See, what is written on mRNA? It will be recognized and read by tRNA. Now, among the tRNA, in the tRNA, which segment is responsible for reading the mRNA? That is your anticodon loop. That is your anticodon loop, and this is your anticodon loop, the one in shaded in blue. So this segment will read what is written on mRNA, and this green portion where there is 3' and hydroxyl group, this is the place where the amino acid will be binding to the tRNA. The pool in the amino acid, the cytoplasmic amino acid pool, out of that pool, one single amino acid will be going and binding to this particular 3' OH, thereby forming a complex of amino acid and tRNA. Now, this particular complex is called as amino acyl tRNA. Amino acyl tRNA. So it's very simple. tRNA is performing two functions: reading the mRNA, binding to the amino acid. The binding is occurring at 3' OH, whereas the reading is occurring at anticodon loop. The reading is occurring at anticodon loop. Now, when we are talking about the protein synthesis, when we had read complete description regarding this tRNA, when we are talking about protein synthesis, how many stages are there? in the protein synthesis? See, primarily there are only three stages if you broadly categorize, like initiation, elongation, and then termination. But if you study in detail, before initiation there is a stage, I call it as pre-initiation stage, or it is the actual stage where the amino acids are getting charged up, they are getting activated to give rise to tRNA molecule, the activated tRNA molecule that is amino acid tRNA. And then the initiation is starting, and then once the termination has occurred, then after termination the nascent polypeptide chain which is formed, it undergoes multiple modifications called as PTM, post translational modification. Once the protein polypeptide chain is formed, it now folds. Now there will be disulfide bond formation. There will be an attachment of glycosyl residue to give rise to glycoproteins. There might be addition of a methyl group. There might be an addition of an acetyl group. There might be an addition of a phosphate group called as phosphorylation, which is required for activation of many enzymes and many proteins in the signaling pathway. So all those are post translational modifications which are happening, of course, after the termination process. So that is the reason why technically we can say that there are broadly three stages, because once the termination has happened, the translation has stopped. Now whatever you are doing with the already formed polypeptide chain in terms of post translation modification. But yes, in this lecture we'll be focusing on activation, then initiation, then elongation and termination. We will not be discussing the post translation modification part, which is happening after termination part. So that's why I have just given you a glimpse of two additional stages that we are talking about in terms of classical stages, in comparison with the classical stages, of course. So when I'm talking about these three stages that are the classical stages, that is initiation, elongation and termination, it's very simple. What is initiation? Initiation in simple layman language means to start. How it will start? First mRNA should come, then ribosome should come, then ribosome should attach to mRNA, then somehow it should bring tRNA there where the first amino acid will come and bind, and then in the elongation the other amino acids depending upon the sequence should come and bind, and ultimately they should be removed from that complex, that is called as termination. So these are the broad categories. Now let us, with all that information, let us now take a dive in the process of translation and understand the chronology of the events, how things are happening. So the first stage in terms of activation, that what I narrated to you, the activation state. What is that? The activation state, see, this is the stage where the amino acids will be getting activated and they will be binding to the tRNA to give rise to amino acid tRNA. Now how that amino acid is binding to a tRNA, I told you the place where it is binding at the 3' hydroxyl group. Okay, so adenine is present at the tRNA, OH group is also present, so my amino acid will be going and binding. But who mediates this reaction? There is an enzyme called as amino acyl tRNA synthetase. Now when I am talking about an enzyme with synthetase, see, there is a type of an enzyme called as synthase, and there is another type called as synthetase. Both of them are synthesizing, both of them are responsible for forming a bond, but synthetase will form a bond with the expenditure of one ATP molecule, whereas synthase requires no energy. That is the basic difference in terms of biochemistry. So an enzyme called as amino acyl tRNA synthetase is present there. Now it will form a bond between amino acid and tRNA. Now let us go and recapitulate the chemistry behind this. There is an amino acid, there is a hydroxyl group which is present at the 3' OH. Now the carboxyl group of the acid, the amino acid, will go and bind to the OH group. Now what is this reaction? Whenever an acid binds to an alcohol, it gives rise to ester. Esters, and that's why the binding of amino acid to tRNA is called as esterification, and this esterification is mediated by the amino acyl tRNA synthetase enzyme, with the expense of energy, of course. And where this reaction is happening? This is happening in cytoplasm. Why? Because the amino acid pool and tRNA both are present in cytoplasm. So what is happening right now? The amino acid is binding to tRNA to give rise to amino-acyl tRNA. Amino-acyl tRNA. Now suppose arginine is binding, then it will be called as arginyl tRNA. Suppose proline is binding, we will be calling it prolyl tRNA. Suppose aspargine is binding, I will be calling it as asparginyl tRNA, which is formed after the addition. So once we have established that what is amino-acyl tRNA, now you should also understand this, that we have 20 amino acids. So minimum what we required is 20 different tRNA. And another thing, that this enzyme, I have written here clearly, amino-acyl tRNA synthetase. Now if aspargine is there, then aspargine specific synthetase will be working. If methionine is there, then methonyl tRNA synthetase will be working. So each enzyme is specific for one amino acid. That's why you need 20 different enzymes for this particular thing. Fine. So you need these enzymes to add the amino acid on this particular tRNA. Now how that addition is catalyzed? What is the reaction that we are talking about? There is an amino acid, there is a tRNA. Now the enzyme will come, and at the expense of ATP, it will form amino-acyl tRNA, AMP and pyrophosphate. But is this reaction so simple, or there are multiple steps? Yes, then of course there are two different steps in this particular reaction. Let me directly jump to the steps. This is the reaction that we are talking about. Mediated by what? Mediated by amino-acyl tRNA synthetase. Now let's see what happens. This is an amino acid. Fine. And this is your tRNA. What tRNA? The last nucleotide, adenine, at the 3' end. Fine. It contains a OH, a 3' OH is present, and also contains a terminal phosphate group. Of course, it contains a terminal phosphate group. Okay. So this particular representation, what I'm talking about, is tRNA binding to amino acid. How it mediates? Let us see. This pink region is amino acid. Now this amino acid will first form complex with the ATP molecule. So the incoming ATP molecule which you are using, this is adenosine, this is triphosphate of this ATP molecule. So we have 3 phosphate molecules. Now what will happen? This carboxylic group will form a bond with the first phosphate which is attached to the carbon, which are calling it as alpha phosphate. Remaining 2 terminal phosphates will be released in the form of pyrophosphate, resulting in the formulation of a compound which contains a anhydride bond, phosphoanhydride bond between AMP, A, that is adenosine monophosphate, because only one phosphate is remaining. So AMP along with amino acid. Now it's a transient stage where amino acid has attached with the AMP. Then what happens next? Now this amino acid is activated. Now this particular amino acid is called as activated amino acid. Fine. Now what is the job of an activated amino acid? The main principle behind this reaction of amino-acyl tRNA synthetase is to take this molecule, that is amino-acyl AMP, take this molecule and attach it on the tRNA. Now where this attachment is happening, it will attach this tRNA on 3' OH, right? So if it attaches to the 3' OH, this is the adenine residue of the tRNA. This is the last adenine nucleotide of the tRNA. Now this sugar molecule contains 2' OH also and 3' OH also. Why? Because this is RNA, not a DNA. In RNA, the ribose sugar contains hydroxyl group or the oxygen group at both the 2nd and the 3rd position. Now, if this amino-acyl AMP brings the amino acid and binds it to the 3' hydroxyl group, then the bond will be formed between the carboxylic group of the amino acid and the hydroxyl group, giving rise to amino-acyl tRNA, and the AMP will be released. So now AMP will be released. So what is happening? The ATP is binding to amino acid, thereby activating it, and it gives out the 2-phosphate. Now the complex which is formed is amino-acyl AMP. Now this will go and and then transfer this amino acid group to tRNA, and in turn the AMP will be released. And this is in nutshell the two-step process that is happening or conducted by amino acyl tRNA synthetase. Now please remember, amino acyl tRNA synthetase takes amino acyl AMP as substrate and never individual amino acid as substrate, until and unless the amino acid is charged with AMP, adenosine adenylate, until and unless it is converted into 5-amino-acyl and adenylate, that is amino acyl AMP, it will never be recognized by amino acyl tRNA, and it will never be attached to tRNA. And then if you are not performing this, if this reaction is not being performed, then of course there is no translation, right? There is no translation. So let us jump to another thing. When we have established that 3' OH is the position where the carboxylic acid is going and binding, where the amino acid is going and binding, giving rise to a amino acyl tRNA. So we have established that the 3' OH is involved. Okay, so this particular axis is called as class 2 type tRNA, class 2 type synthetase enzyme. But the enzyme that we are talking about has its variant also, that is called as class 1. Now what this class 1 is doing, that's the most interesting part of this reaction. I told you that this particular sugar group at the terminus of the tRNA contains both the oxygens, both the hydroxyl groups at 2' and 3'. Now the addition of amino acid depends upon what type of enzyme is present, whether it is class 1 enzyme or class 2. If class 2 is present, then of course 3' hydroxyl group will be in the part. But for class 1, the bond is now formed between 2' OH and carboxylic group, giving rise to again amino acyl tRNA, but the attachment is at 2' OH. But what we need for translation, please keep this very clear, we need 3' OH attached amino acid if we want to talk about translation in ribosome. So this particular amino acyl tRNA, which is associated with 2' OH, is of no use, but it never goes waste. Why? Because after formation of 2' OH amino acyl tRNA, it gets converted back to 3' OH tRNA by a process called as transesterification. So you see, whether it is class 1 tRNA synthetase, whether it is class 2 amino acyl tRNA synthetase, eventually they both will be landing up to giving you a same product, that is 3' hydroxyl group attached amino acyl tRNA. And this marks the completion of the understanding of formation of amino acyl tRNA. So you see, eventually it is culminating to a single point where there is a formation of amino acyl tRNA. Now there were multiple things that has happened. Initially there was amino acid pool, it got charged, it got activated, it got attached to tRNA by a certain set of enzyme, and depending upon the type of enzyme, it got attached to 2' or 3', and thereby giving rise to 3' amino acyl tRNA. Once this particular accomplishment has been done, fine, once the activation of amino acid has been done and the attachment of amino acid has been done to the adapter that is called as tRNA molecule, fine, then this particular identity of the amino acid which is attached to that particular tRNA, fine, then this tRNA along with the amino acid, this will determine the fidelity of protein, because this is a molecule which is actually responsible to bringing the amino acid. If somehow this adapter molecule is bringing a wrong amino acid, then eventually there will be a mutated protein or a truncated protein which will never fold, right? So this particular path marks the error free. If this particular process step is error free, then of course the protein synthesis should be error free in terms of translation machinery. Then let us go to the stage 2. Now what is stage 2? In stage 2 I am talking about initiation. Now we had already established pre-initiation. Now let us talk about the initiation. What is happening? So first of all, please be clear in your mind, when I narrated about DNA replication, the formation of new strand, that is DNA, was taking taking the direction of 5 prime to 3 prime. mRNA was also being synthesized in the same direction. What is the direction of synthesizing a protein? Now there is no 5, no, there is no 3. There is only N terminal and C terminal. The first amino acid is now here. Fine, the first amino acid is here and the incoming amino acid will be attaching here. Okay, so it is attached in what manner? The carboxyl group will be available for forming a peptide bond, and the opposite side there will be an amino group, the N group, the amino group. The new amino acid is coming and forming a peptide bond. So two amino acid is now joined. Fine, two amino acid is now joined. Now this will be joining, forming the peptide bond with N terminal amino group and the carboxyl group. So that they have joined. Then again a carboxyl group is opened for the incoming amino acid. So what is happening? What is happening? We are moving from N terminal towards C terminal. So this is the direction. This is the direction that protein synthesis always occurs from N to C terminal. N to C terminal. Then the starting codon that we talk about, initiation codon, is always methionine, that is AUG. And while studying the genetic code, we have clearly established that AUG is the only codon for methionine. Is the only codon for methionine. Fine. So when the initiation is starting, when the mRNA, the first amino acid which is added always to any kind of protein is methionine. So the methionine tRNA is responsible for starting this particular translation. Now what kind of methionine? What kind of methionine? Let us see. Now see, for methionine there are two type of tRNA which are present inside the cell. The one is which is only specific for the starting codon, at the starting position where the methionine is present. But suppose during the complete length there is another methionine residue which is to be incorporated in between. For that there is a separate tRNA. So these are the two types of tRNA. One tRNA will be specific for the start codon, and the other will be specific for the methionine which is being incorporated in somewhere in the internal position of a growing polypeptide, Now when I'm talking about the initiation process, please understand clearly what these two tRNA are there. See, in bacteria or on the prokaryotes we are talking about, the two tRNA of a methionine are designated as tRNA and Fmet tRNA. One is normal methionine tRNA, methionine tRNA. Another one is Fmet, formilated methionine tRNA. Now this Fmet is the tRNA which recognizes the start codon. So the first amino acid for the protein in prokaryote is always formilated methionine. Fine. So what is happening? The amino acid which is incorporated at the first starting in response to the first codon, that is AUG, is always formilated methionine, N-formyl methionine. Fine, N-formyl methionine, which is brought about by formilated methionine tRNA. Fine. So how this process is going about, whether it is directly attached or how this particular formilated methionine tRNA is formed. So what is happening? First of all, what is happening? The methionine tRNA synthetase enzyme, what it does, it attaches the methionine to tRNA. After the methionine is attached to the tRNA, then a separate formyl group is attached to that particular methionine by it is attached to the methionine by this particular reaction. If you clearly see, first there is a methionine which is getting attached to that tRNA, the tRNA which is responsible for this for responding to the start codon at the expense of ATP, giving rise to amino acyl tRNA, that is methionine tRNA. Now at this methionine tRNA to the methionine, a formyl group from formyl tetrahydrofolate will be transferred. The tetrahydrofolate will be released, and this formyl will be transferred to this particular methionine, giving rise to formilated methionine tRNA. So this is the tRNA which is actually responsible for the initiation process, And you see how these things are going about. Once the formulated tRNA is ready and we have understood how it is formed, then what are the different steps that are, what are different players that are we will be talking about in this particular initiation in bacteria or in prokaryote. See, we require 30s ribosomal unit. We require, first of all, we require 30s ribosomal unit. This is the chronology of the events, right? First of all, 30s ribosomal unit. Now in that 30s we have 10 different protein and 16s rRNA. Okay, then the mRNA will be coming, the mRNA which is coding for the polypeptide, that will be coming, that is required. Then the initiating tRNA which will bring the formulated methionine, that will come. Then along with these three we require three different proteins called as initiation factors, IF1, 2 and 3. These are called as initiation vectors. We'll see what function they perform, but these are of utmost importance for initiating the correct initiation. Then we also require a JTP molecule which will be responsible for releasing the energy. Then after this, after all these have come, then the larger subunit is now coming. The larger subunit will be required, the 50s, to give rise to a complete ribosome, the 50 and the 30 giving rise to 70s. And we also require a cofactor, that is Mg plus 2 ion, Mg plus 2 ion. Then first let us see what functions are being performed by this initiation factor. What function is performed by this initiation factor? The IF1, IF2 and IF3, what it is doing? See, let me just give you a picture how this site is looking. Let us first focus on this picture, then we'll again go back to the previous slide. See, this is the 30s subunit, the small subunit. It contains two sites, the A site and the P site. The A site is called amino acyl site and the P site is called peptidyl site. Okay, the P site is called peptidyl site. Now while understanding the functioning of these transcription factor, I'll be referring to these two sites. Okay, so what functions are performed? See, IF1 goes and binds to the A site. Why? Because if it is blocking the A site, the incoming first tRNA, that is formulated tRNA, will be having the only single option to bind to the P site, and P site is a place where the initiation will be starting. So in order to prevent the non-specific binding of tRNA to A site, this initiation factor 1 is of utmost importance. Then what about IF3? Now this IF3 will be going and binding to the small subunit, that is 30s RNA. It will bind to the 30s subunit and it will prevent the binding of this small subunit with the large subunit. Why it will prevent? It will prevent because it will keep this on check until the complete initiation complex is formed. The tRNA has come and bind to the P site, the mRNA is already there and everything is now ready. We need now to add this particular 15 ribosome unit, then only it will be detached. Until and unless the complete initiation process is ready, initiation complex is ready, IF3 will prevent the binding of these two ribosomal subunits. Fine. Then what about IF2? Now this is very crucial. IF2 initiates promotes the binding of formulated methionine to the P site, the actual site. You see there are three factors. First is binding to A, preventing the non-specific binding to A site. The second is binding to P. It will promote the binding of formulated to P site. And then there is another IF3 which is binding to the ribosome. It is preventing the binding of small subunit with the large subunit until the replication is formed. And these are the functions of your initiation factors. Now let us see how these functions are being carried out. Now you see here, there is a small subunit, there is P site, there is A site. Now first of all, the initiation factors will come. IF1 will bind to A and IF3 will bind to small subunit, and this marks the onset of translation. And then the mRNA molecule, which is actually responsible for bringing the information, will be coming and binding to this small subunit. Now whenever we will be reading, when we complete the reading of this particular translation process, we'll be talking about three sites, A, P, and another is E site, the exit site. Now E site is contributed by the large subunit, so that's why we are not showing here. Let us go step by step. Okay, let us go step by step. Okay, so when this factor IF3 is binding to the small subunit, it is preventing premature binding with the larger subunit. Fine, and then the mRNA which is coming and binding to this small subunit. So at what position should this particular be binding taking place? Now there is a sequence that is present called as Shine-Dalgarno sequence. Shine-Dalgarno sequence, which is present upstream in this region, upstream of the initiation codon in mRNA. This Shine-Dalgarno sequence is present in the mRNA, which acts as a guide for correct positioning of 16S RNA, so that the first codon should be placed in P site. The first codon should be placed in P site. That's the function which is performed by the Shine-Dalgarno sequence. The Shine-Dalgarno sequence will be the site where the 16S ribosome RNA will be binding. This particular 30S subunit will be binding small subunit, and then it will be placing itself in such an orientation that the AUG, the first codon, is placed at the P site. And this is how the complete process is brought about. This is how the complete process is brought about and mediated by the Shine-Dalgarno sequence, where you are actually putting the first codon, that is AUG at the P site, where the formilated tRNA, formilated methonyl tRNA will be coming in binding. Now, when the formilated methionine tRNA, which will be binding to the P site. Now, please remember, out of all the tRNAs, out of all the amino acid tRNA, only and only F-methonyl tRNA is the tRNA which can come and bind to the P site. Rest of the tRNA will be binding always on the A site, never to the P site. But this first one, the first tRNA comes and binds to the P site, Okay, now this particular binding is facilitated by initiation factor 2, by IF2. Okay, this is initiated by IF2. Now once this tRNA has come and bind, now the complete complex is made up of what? Let us see. Now the complete complex is going about what? See, once this upper picture is over, then the next picture starts. What is the next picture? The formilated methonyl tRNA along with IF2 is coming and binding to P site. So please remember, IF2 never binds to the small subunit. It binds to the incoming formilated methonyl tRNA. By binding, it brings the first tRNA molecule to the site, that is P site, at where the first starting codon is present, that is AUG. Now what is happening? This initiation factor IF2 is not alone. It is always bound with GTP molecule. It is always bound with a GTP molecule. So at current state, where IF2 has brought a new tRNA molecule, that is formilated tRNA molecule, to this particular P site, what constituents we have? In this particular complex, we have formilated tRNA, formilated methonyl tRNA, we have mRNA, we have small ribosomal unit, we have IF1 at A site, IF3 with ribosome, and GTP bound IF2 along with tRNA. This is now ready for what? For allowing the larger subunit to bind upon, and let's carry out the translation. So what happens next? In the step 3? In the step 3 now, this particular larger subunit, that is 15S ribosomal unit, will be now binding to the small subunit, and thereby completing the ribosome. And what happens then? Once this particular 50S subunit is coming, the GTP which was present on IF2 is now hydrolyzed, and the energy that is given by this GTP is used for the binding of this larger subunit. And once the binding is achieved, now all the 3 initiation factors will be released. They will be departed from that particular site. Now this particular complex is called as initiation complex. Now the formation of initiation complex is complete. Now this complex contains both the units of ribosome, A site, P site, which is in small, small subunit, and the E site called as exit site at the large subunit, and at the P site to the first starting codon AUG there is an attached formilated methynyl tRNA. Now what happens next? Initiation is done, or is there something else that needs to be done? See what will happen next. Now the next codon will be read, and the next tRNA will bring the amino acid and bind to the A site Now that process is called as elongation. Now the elongation is starting. See what is happening in the elongation process. Now there is a vacant A site. Now what, already a formilated methionine is present. So whichever amino acid needs to come, now it needs to come to the A site in order to form a peptidyl bond. Fine, and that bond once formed marks the starting of this elongation process. So what is required? The initiation complex should be ready. In terms of elongation, if you want to elongation to happen, the initiation complex should be ready. Then all the amino acid tRNAs should be ready. They should be available in order to bind the amino acid. Then another 3 sets of proteins called as now elongation factors. Now these factors will be responsible for elongation process, and then along with them energy will be required in forming every peptide bond, and that energy is given by a GTP molecule. Now if we talk about the number of steps, so cell takes around about 3 steps for adding every amino acid, and then these 3 steps are repeated every time for one amino acid when it is being added. So let us see what are those steps. The step 1, if we talk about, see the step 1, what is step 1? Our initiation complex, pre-initiation complex is ready. Now the next amino acid tRNA is coming. The second amino acid is coming bound by this particular tRNA. Now first of all, the elongation factor TU, EFTU along with GTP. The EFTU is GTP bound elongation factor. It will bind to that particular amino acid tRNA. Fine, it will bring to the particular A site. It will bring the particular amino acid tRNA to the A site. The amino acid tRNA will be attached to the A site, and the GTP will be utilized. Or hydrolyzed in order for this tRNA to get attached to the A site. Now once this elongation factor has delivered the amino acid tRNA to the A site, it is released, and then again it acquires the phosphate group and then converts itself into an active elongation factor EFTU bound with GTP, and thereby it can again bound to the other incoming amino acid and can channelize the delivery process. So it's a cyclic process. It binds to a tRNA, amino acid tRNA, it brings releases the GTP or hydrolyzes the GTP, releases the energy, delivers the tRNA, then again go back, activates, attains the GTP, and then again the cycle is repeated. So you see elongation factor, what it is doing, EFTU, what it is doing. It is actually mediating the transfer of amino acid tRNA required for the growing chain, at the expense of energy that is given by GTP, and that's what is happening here. Then what happens the next step? See what has happened. EFTU has done his job. It has delivered the new second amino acid bound with the tRNA at the A site. Now what is the job? Now the job is to form a peptide bond. We need to form a peptide bond so that amino acids can be joined to form peptide. The amino acids are shown in blue color. The bond will be formed between the P side and the A side. The P side's carboxylic group will be forming the peptide bond with the nitrogen or the amino group of the A site. That's why I told you the N will always be free and it is moving towards N to C. So carboxylic group of the first residue and the amino group of the incoming amino acid forms a peptide bond. And that's how the reaction is from N to C. Now this particular peptide bond is formed by an enzyme called as peptidal transferase. Is formed by an enzyme called as peptidal transferase. Once this particular bond is formed, once this particular bond has formed. Now, after formation of this bond, what happens? After formation of bond, what happens? After forming a peptide bond, the amino acid formylated methionine is now transferred to the incoming tRNA, and this particular tRNA is now vacant. This particular tRNA is now vacant. So what is happening? A dipeptidyl tRNA is formed at the A side. A dipeptidyl tRNA is formed at the A side. What has been done? The formylated methionine has formed a bond with the incoming amino acid, and then because of the formation of peptide bond, this particular amino acid is released to this particular A side tRNA. Now the initiator which was responsible for starting the initiator is become vacant. Now this tRNA is vacant, which is present in the P site. Now what is next? What is next? Now next is this particular dipeptide which is present at the A site, and the vacant tRNA which is present at the P site will see a translocation. Now this is the third step. What is translocation? See, this is what we had studied right now. Now additional elongation factor will come. Now an elongation factor will come called as EFG. This is again a GTP bound elongation factor. Now translocation will happen. Why? Because another amino acid needs to come and join, and there is no vacant site. So we need to create a vacant A site. So what will happen? This ribosome will now move. mRNA static. This ribosome units will now move, and this movement is called as translocation. How it moves? If it moves from left to right, so what will happen? The vacant tRNA will enter to A site. This dipeptide will be aligned on P site, and then again A site will be vacant, and then again A site will be vacant, and that is what is happening here. The translocation has taken place. The vacant has come on A site, the dipeptidal has come to P site, and the A site is now vacant for the another incoming amino cell to come and form a peptide bond, and then there will be tripeptidal, tetrapeptidal, and so henceforth, so on. The chain will grow and continue. And this translocation process, this translocation of ribosome is energy dependent, and that is mediated by elongation factor G, EFG. That elongation factor brings a GTP molecule and hydrolyze that particular GTP molecule, and then moves this particular ribosome in right direction. And that's why this elongation factor G is called as translocase. So you see what happened. A initiator which was present, incoming amino cell tRNA, they formed a bond, and then suddenly what happened? The ribosome moved, and now this site is vacant. Now another amino acid can come and bind. So what is happening? The bond is formed. The elongation has started. Fine. And that is how the complete dipeptidyl tRNA and the GTP molecules are utilized. So you see what is happening here. For every amino acid that is being added, there are two GTP molecules which are hydrolyzed. One was with EFTU in order to bring the amino acid tRNA on the A site, and the other by translocase EFG in order to shift the ribosome to create a vacant A site. So for every addition, please remember, for every addition of an amino acid to the growing peptide chain, two GTP molecules are required. How many GTP molecules were required in order to initiate? I leave it up to you, because at the end you should be able to answer how many total GTP molecules were utilized in protein or translation. Fine. That is the beauty of reading this particular translation process. Why? Because it is highly coordinated. And then let us see the last phase, called as the termination part of a polypeptide chain. How the termination actually is going about, how this termination is happening. So see, like the starting part, we have a start codon which was specific for starting. We already know, as per our understanding of the genetic code, that we have three stop codons in the mRNA, that is UAA, UAG, and UGA. These three codons act as stop codon and acts as terminators for this particular polypeptide synthesis. for this particular polypeptide synthesis. So the question is, in bacteria, if we're talking about in a prokaryote, is this a spontaneous process, or this is mediated by different types of protein factors? Then of course, again, in this phase, the termination phase, there are again three factors which are involved: RF1, RF2, and RF3. Releasing factor 1, 2, and 3. Releasing factor 1, 2, and 3. What they contribute to? They contribute to the hydrolysis of the terminal peptide tRNA. The last peptidyl tRNA bond that requires to be bound, they cause its hydrolysis. They cause its hydrolysis, and thereby they release the free polypeptide. They release the free polypeptide, and the last tRNA which is left will always be uncharged from the p site. Let us see what happens. Let us see what happens in this particular section. See, the chain has grown to a certain length, and now a stop codon has come to a site. As soon as the ribosome moved, the A site is now vacant. At that vacant site, it encountered a stop codon. Now, as soon as it sees a stop codon at A site, the releasing factor comes and binds to this particular A site. The releasing factor will come and bind to this particular A site. Now, this releasing factor will recognize this particular stop codon and will cause the hydrolysis of the ester bond. Which ester bond? The bond which is formed between the amino acid and the 3' OH of the tRNA molecule. Amino acid and the 3' OH of the tRNA molecule. So you see, amino acyl tRNA bond which was formed, now it is being released. But by the release, what is happening? The complete chain which was present at the P site is now released. Now the protein is released. The complete sequence of the protein is now released. The amino acid chain is released, and RF has brought about the termination. Now what happens next? This tRNA, the uncharged tRNA, remains in a bound state at the P site. The chain has been released. Now what will happen? This release factor will cause the dissociation. First, it will cause the dissociation of the subunits. The large subunit and the small subunit will be dissociated, and then the RF and the tRNA are now released, and the mRNA is free. And then summarizes your termination process. Then what about the release factors? The RF1, there are 3 codons. The RF1 is responsible for UAG and UAA. The RF2 is responsible for UGA and UAA, the stop codons. It recognizes different stop codons, but essentially they catalyze only 2 functions. What functions? They first stop the binding of any incoming nucleotide on the A site. Second, they hydrolyze the ester bond, thereby releasing the protein chain, and the uncharged tRNA remains bound to the P site. Then they dissociate the subunits, and voila, your complete process is now over. The mRNA is free. The mRNA is set free with the process, and the complete protein chain is formed. So you see what we have done till now. Let us see. Let us summarize in just quick 1 to 2 minutes. There were 4 steps that we discussed. The pre-initiation formation, where the activation of amino acid happened, the initiation, elongation, and termination. The activation part, where the amino acid is going and binding to the tRNA, mediated by amino acyl tRNA synthetase enzyme. It is binding the amino acid to a 3' OH of the adenine nucleotide that is present at the 3' position. Depending upon binding, we saw 2 different class 1 and class 2, right? So the tRNA is now activated. Once it is activated, then the initiation starts. How the initiation is going about? It is mediated by initiation factors 1, 2, and 3. How those factors are working? Those factors are working by binding to the small ribosome unit. The IF1 is binding to A, the IF2 is binding to P, the IF3 is responsible for binding to small subunit. The IF1 binds to A site so that it can prevent the incoming non-specific binding of tRNA. The first initiator codon, that is AUG, is recognized by a formilated methonyl tRNA, and that formilated methonyl tRNA is bound with IF2 and GTP complex. This complex will be guided to P site, and IF3 is responsible for not allowing the large subunit to bind, right? Once the FMET tRNA is bind to P site, now the initiation factors will be released and the large subunit will bind. Once the large subunit is bind, then the elongation process will start, and then the new -L tRNA is coming at the A site. Now there is a peptide bond formation in the elongation process between the amino acid in the P and the A site. Once the peptide bond is formed, Then there is a dipeptide bond formed, and there is a only tRNA which is present on the P side. Then the translocation process has happened. The translocation has happened, creating a vacant A site, and that's how the elongation appeared. And then we're talking about the termination in contribution with the releasing factors, along with the releasing factors. So once the releasing factors have come, the RF1, the RF2, the RF3, all three of them performing the same function. What function they are performing? Just to release the polypeptide chain, and then dissociate the complete system. And this is how the complete prokaryotic translation is happening. So also remember one small thing in the last what I want to tell you. See, this is a prokaryotic model, and please have a clear notion that this process of translation is happening simultaneously as the mRNA being synthesized, because there is no different organelles. In prokaryotes, mRNA is synthesized, and side by side the same mRNA is used for translation process. So translation is happening simultaneously with transcription in case of prokaryotes. So let us summarize it with certain kind of MCQs, because it is required to brushing up. P site on the bacterial ribosome is known as what? Is it a peptidal, polar, what kind of site? When you're talking about the energy rich part, which energy molecule is required in this complete synthesis of protein molecule, whether it is ATP or GTP? Which of the following RNA molecules is actually an adapter molecule? Or if you're talking about the stop codons, what are the stop codons? Or the E site is present in which subunit, whether we are talking about the large subunit or the smaller subunit, and which is a large, which is a smaller subunit in the prokaryote, you should always remember. Then what was the first codon that I'm talking about in terms of prokaryotes, and the process by which protein synthesis is being done, what is the technical process? Whether we are talking about when we are talking about the translocation, what exactly is translocation? Is the movement of tRNA molecule, or it is a movement of ribosome, or it is the movement of tRNA from A to P, or P to A, or P to E? What is the chronology of the sequence of the event? And when you're talking about the polysome, what are polysomes? So these kinds of questions, when you go through the literature, will be summarizing your part. And for your future reference, I've just listed few of the articles that are beautifully published by different researchers across the globe, different narrators across the globe, and it will help in your clear-cut understanding of this particular topic. So I hope the complete lecture was fruitful to you. You must have understood the chronology of the events and how this particular translation process is taking place inside a prokaryote. With this, I'll thank you for your kind attention, and I hope we'll meet again in the next lecture with another different topic. Next time we'll be discussing translation in eukaryotes. Till then, please stay safe and wish you a very, very happy learning. Thank you so much.